I'd like to call to the stage Professor Raul Urrutia, Director of the Linda T. and John A. Mello Center for Genomic Sciences and Precision Medicine at the Medical College of Wisconsin. Raul, thank you very much.
Thank you so much. I would like to congratulate Professor Ifra, Professor Mano, Your Excellency Ando. I would like to thank very much to the organizer, Professor Joanna, for this invitation. And I know that Dr. Cashnell has also played a significant role in the organization of this meeting, which I think is very, very, very timely. Josie, we need more of you. Today, we are having a large representation on scientists. We have tried. We are trying. We are more involved in the revolution like you, but we need more like you. Thank you very much. The Linda Mello Center for Precision Medicine has a long history that I would like to bring to you. It was the first center in the world to do the first genomic screening with whole exome sequencing. And so it was the launching of precision medicine and received the recognition of many presidents from all over the world. And so that tells you already that we are not in the resignation, we are in the revolution. And so I hope that I can take you through that revolution. So this has been shown by Joshi. I don't think I will go more in detail, but actually the prognosis is very bad. We think that it's going to become the second cause of cancer of the in the United States and around the world. Now, I want to show you this small slide because I started in the 1980s doing pancreatic cancer research. Yes. My name is Raul Urrutia, and I started when I was 18 years old doing pancreatic cancer research, and I remain doing pancreatic cancer research. And when I started, this was the incidence, and you can see that the incidence has increased, but it has increased also because we have better detections. And the mortality rates actually compare more or less with the incidence. So this story, for me, who has seen the development of all the animal models and all the things, is very inspiring because it tells me that the title of this conference is clear. We need a revolution. And to talk about revolution, I will bring 1962, an epistemological work by Thomas Kuhn, which says that we don't change science by just doing papers. We can publish 150, we can publish 300, we can publish 1,000 papers, we can publish 10,000 papers. But science changes by the change in the paradigm. So one day we thought that the Earth was at the center of the universe, of our solar system. But then the calculations were not given right until we put the sun in the middle of the universe. And when we put the sun in the solar system, and now you're seeing system biology, all the space started to make sense. So we would like to bring two epistemological approaches that have changed throughout the years. I started with the epistemological approach of reductionism. This is the dark of Pascal. If we cut the tumor 1,000 times, we're going to know more of the tumor. Maybe not. And this is system biology. This is where we are. I am a system biologist, and I want to explain to you what is system biology by looking at the brain rather than the pancreas. So if we are taking the brain with the reductionist approach, we're going to cut it all completely. We can mush it, we can liquefy it, we can do anything. Now, my question is, are we going to understand love? Sadness? Human desire? No, because that is an emergent property of the system. It's not any of the little parts put together. And it is this, what is called emergent properties, is that we are looking for. And I know that there are many people here in the audience, colleagues of mine that are also in this revolution. That's what I'm making this introduction. So precision medicine has brought to us a historical moment. This is the revolution with the development of human-derived models and AI applications. We have the power needed for system biology now. And that has been a change in paradigm from reductionism. So we can do not only genetics, you will see how revolutionary also I am, because it's not only genes, right? We can do proteomics, we can do lipidomics, we can do many different things and put together, and what is the emergent property of that system that tell us. But of course, what we are focused is in the patient. Now, this is our group, and the first thing that the paradigm has changed is there is no one person anymore. This is a team science. We do from atomics models and simulation, protein phenotyping, genetic engineering, cellular models, human-derived animal models, and experimental therapeutics. And we look at the systems, and we just actually work with all those systems of the cells in order to look for the emergent properties. And I'm going to show you work that it has been that way. So in our unit, we have a translational data science team and a translational experimental team, and both of them work together. We have a board, we have omics, multi-omics, we have computational biology, you will see. We have a structural biology that we need very much, and we have the multi-omic unit. And I'm going to show you the first revolutionary paradigm from our laboratory many years ago. And it was when the Wolkenstein paradigm was very active, and I was very young. And one of the things that I could not believe is that the cancer was going to be genetics. Now, I know that cancer, like Yossi, was inherited, and that genes play a big role. But there was another part of inheritance called epigenetics. And so we said, how are KRAS talking to the epigenetic mechanisms, let's say, in order to do pancreatic cancer initiation? So already we went from the genetic-centric paradigm, right, extending it toward the epigenetic mechanism. And what we realize is each time we touch the epigenetic mechanisms, we change the nuclear structure. And if you see the change between panning 1 and panning 2, it's not a large amount of mutation, but the change in nuclear structure. And that's one active area of study in our laboratory. So our friends here in the room started with a large amount of experiment. We did too. And some groups came out with the genomics, and we have learned a lot of the genomics. But so far, we knew that there was only one pancreatic cancer. Because why? Because the genomics, as I said, is one part of the story. And from genomics, we could not differentiate any subtype of pancreatic cancer. Other colleagues, however, came with something more interesting for me. They showed that the signatures, either in RNA-seq or in proteomics, was given subtype. And I can read from the RNA-seq what it could be happening at the epigenomics. So when I saw this, it said there are 2 epigenomic subtypes. So we went to find the mechanisms, and indeed we use Avatar, and we use like 9 different omics in order to come out with the fact that pancreatic cancer progression is not completely genomics. As a matter of fact, it's primarily epigenomics, just like the differentiation in the embryo. And is the result of enhancer and super-enhancer. So there is a classical and there is a basal cancer, right? And we found that the basal cancer is more malignant, and it also has some oncogenics like the c-MET oncogene driving it. While the classical is more like embryonic-like. It's more differentiation. Unfortunately, it's the first one that is killed by chemotherapy. And so what we published there is that regardless how it starts, and this was a good time in my life because I have been since I was in my 20s saying that epigenetic has to play a role. So that was the revolution, Josie, that whatever was starting here and all the cumulative mutations, there was a predestination to become into these two types. More importantly, and this is a molecular experiment, no human experiment, we took cells and we knocked out the c-Met driving, and we were finding that basal could transform into classical. This was before the mixed tumors were found, and we predicted with this the finding of the mixed tumors that do exist. So the conclusion here is that pancreatic cancer is a heterogeneous disease, which we didn't know before. It's driven into progression through epigenomic mechanisms and alterations in enhancers and super enhancers. In fact, we have developed some therapies against enhancers and super enhancers to actually try to fight this disease. Now, by that time, that was progression, when the cancer has progressed. And we said, what is happening with initiations? Well, we took KRAS G12D, which is the most famous oncogene, and we did all these system biologists, omics dimensions. So we did ATAC-seq, we did CHIP-seq for 5 different markers, enhancers, promoters, gene bodies, etc. We did RRBS methylation, we did RNA-seq, several chromatin peaks and studies, and then we did the integration. Now, what I'm going to show you is that there are 2 integrations here. One that that is here. These are the super enhancers, and so what it says that from the state 1, KRAS, off, to this state, which is KRAS signaling oncogenically, the most important change is enhancers and super enhancers, just like in the progression of pancreatic cancer. Now, of course, this was G12D, only one of the KRAS, and there are many. So what we did, let me see. I have a problem with this. What we will see is we applied another paradigm. And you see the importance of the paradigm using rather than going anywhere to look for. This paradigm says that KRAS will actually signal, will not only signal to the nucleus to change, but it will also signal to the tumor microenvironment. I call this the smart city because the oncogene single-handedly can turn on its own microenvironment. And we have found out, and I will show you, is that KRAS regulate the microenvironment very heavily. And what we did is first we took 25 distinct KRAS mutants, the most common 25 mutants of KRAS. We do the structural biology. You can see that they are heterogeneous. In other words, there is heterogeneity in KRAS initiation. We did enzymatic activity, we did pathways and all the signature, and what we have found out is there are groups in KRAS initiation. Structurally, we started with one, and this was work I did with my hand in order to see how G12 was behaving, and then we went to 88 mutations, and now we go to 1,000 mutations. Of all the RAS families in the different cancers that the RAS actually work. And we increase the scale. We are now actually able to do 5,000 mutations without any problem. Now, we also have this group, which is the SGU unit, the structural genomic unit. And here we purify proteins, like you see here, and we do intrinsic and extrinsic exchange activity of the oncogenes. We also do NMR fingerprinting, and what you can see here is how the different atoms move, and then we project in order to see whether the KRAS are structurally different among themselves. We've done this with 65 KRAS, and what you see is that they have different thermostability, and yes, indeed, they have different shape. So they are different, they are heterogeneous among themselves. 65 KRAS heterogeneous. So we did the first study biologically, right? We said, what is doing in the cell? So the first work I'm going to show you is we did it in HEK cell because we wanted to do in something that was a platform for genomic, very useful, which is HEK cells. And we put 25 different KRAS mutations. The most famous 25 mutations. And again, we did system biology in order to see the emergent properties. I'm going to show you this. You don't need to appreciate much difference, too much, the slide to see the differences. This is ERK, and you can see how the different KRAS are heterogeneous in ERK signaling. This was the first work we have done now with large amount proteomics, and they are really heterogeneous in protein expression. They are heterogeneous also in transcription, but they form groups, okay? They form groups. So that's the good news, that some of them are more similar than others. And there are approximately somewhere between 4 and 5 different groups. And you can see, if you measure G12D here and you look at the similarity to G12D, you see the entire heterogeneity that they have in expression. Now, obviously, we are pancreatic cancer, so immediately we actually kick the most of the best known pancreatic cancer oncogene, KRAS, in HPNE cells, and then we did the same experiment, and what we found is that there is heterogeneity in the transcriptome, which, by the way, is different than the heterogeneity in the other cell, because the epigenetics of each cell is different. So this is somewhat applicable also to colon, but it's more specific to pancreatic cancer. And so you see the heterogeneity here. You see how they are heterogeneous. You see the heterogeneity in the pathways. And this is proteomics, okay? And in proteomics, we look at immunosuppressives, immunostimulatory, cell survival and response, and you see the different KRAS and the heterogeneity among them. This is another way of doing what we did here is large-scale ELISA, and we measure all what they were secreting, and what we found is that they are powerful regulator of the tumor microenvironment, but they are heterogeneous. For instance, here, there are all group here, H61Q is out there, G12R is here, but if we take these two, then we also see the heterogeneity here. So that there is a heterogeneity in the tumor microenvironment as we know it, because KRAS does lesion in different organs with different tumor microenvironment, okay? So there is heterogeneity in there. This is very important because there are many things that are coming out for fighting in a tumor microenvironment. Obviously, we are looking at the epigenomic of this as of now, but we know very well that they are regulated by these 3 complexes, this heterogeneity. The nucleosome remodeling that is working by Dr. Zimmerman in the laboratory, the repressor complex of the repressors, which is Dr. Lomberg, and I work on the compass complex, which is the regulator of enhancers and superenhancers. So, pancreatic cancer progression is a disease of enhancers and superenhancers, and pancreatic cancer initiation is a disease of enhancers and superenhancers. So we have epigenetic then at the center of the paradigm. Now, I'm going to show you very fast, because I have very little time left, what we are doing with computational phenotyping of the epigenomic regulator commonly mutated in pancreatic cancer. So this is, again, we are focusing on enhancers and superenhancers. This is KDM6. And mutations in KDM6, we have actually looked at 136 mutations. These mutations are responsible for a female-enriched pancreatic cancer. And we are very interested in this tumor because it's an epidermoid tumor. It's different than the other tumors. And these females do bladder cancer and also pancreatic cancer. Now, this enzyme is the one that activates superenhancers. It will take the 3-methyl-K27 for P300 to actually activate the enhancer. Now, what does it mean? Because for us, we do all this revolution, but Josie, we want to come there. We want to come to the patient. This is there is somewhere an invitation somewhere from Anirban Maitra there, but we wrote this, which was mutant KRAS inhibitors enter the scenes of precision therapeutic to pancreatic cancer. We are excited. You can see Anirban every day with great news about this. But what our work tells you? Our work is telling you that we have to fight a little bit more. Because our data predict that because of that heterogeneity, the mechanism of resistance and the mechanisms of effectivity are going to be different. So again, it's the word that Yoshi said, that partnership between the GP and the entire team. That's the revolution. Science is not a one-person act, it's a team. And so the future of precision medicine, where we have done taking tumors, we do a large amount of manipulations, but the future of precision medicine is in invent, miniaturize, engineer, use AI enhance, and anything that we are doing in the tumor trying to focus on the patient. Okay? In other words, we have to try to bring the resolution that we have today of the emergent to the patient, and what I propose is that we develop not only a scientific-centric approach, but above anything else, a patient-centered approach, and that is where I see the revolution. That approach should have, as Joseph tells us, mind, body, and soul. And my last word is I love the title of the convention, from resignation to revolution. I do not know much French, but I would like to say that je suis révolutionnaire. Thank you very much.
Professor Urrutia, thank you very much. Told with such passion, technical and detail, so much to take in, but so much passion. And I'm sure there are lots of questions. And the holistic approach, mind, body, and soul, that really stands out at the end of that presentation. Don't forget your questions. We're taking questions from the floor, also online. So don't forget to tap those questions into your phones. Also raise your hands if you have a question. You were passionate. I said you were passionate there. The holistic approach, mind, body, soul, that stands out for me. But I'd like to know, what are you most excited about at the present time? Where is the most promise? When we look at all of this, what's exciting you at the moment where we see the best possibility for a major advance?
Yeah, I think that the easy answer for me would tell you, would be to give you 6 or 7 scientific answers to this question. But the only revolution I've seen in the next 20 years, and the larger revolution, is the one that Yoshi showed you. I don't think that we scientists alone can do the science and get the population to come for us to cure them. I think that the next revolution is actually, again, as I said, patient-centric. This has happened with PanCan, this has happened with Lutz Garden, this has happened with Marjovic. There is a large amount of movement. And so it is the humankind who is trying to fight with us. For us scientists, that is revolutionary, as I said in French, because it is something that didn't used to happen before. We were in the ivory tower. But Yossi, for instance, can hug with Dr. Maitra because they know for many years. So we work together, and that's the revolution.
And when you said we need more of you, Yoshi, is that happening? Is that happening enough?
It is happening. It is remarkable. I mean, I actually, in the last 20 years, I've seen, let's say, PanCam coming from a single person doing some supporting to become, and I'm telling you only one, PanCam, right? There are many, and I respect all of them. But this is happening. We are talking to the government. This year, unfortunately, the United States is going through a terrible time. They have cut most of our research for pancreatic cancer, as it is not deemed to be as frequent as other cancers. We have lost, many of our colleagues has lost that. And so I think that this conference is emblematic of the spirit that we have to have. Scientists, we have a country, but nation, no, we are international. So scientists should help other scientists, and I think that with the help of individuals that are advocate, we can get to those people because, hey, I grew up in a very, very rural, poor area of Latin America. What Josie was saying, individuals sometimes come with an ulcerated breast cancer for the first time without knowing what it was. And so I think that there is a lot to do getting closer to the patient.
Okay, big challenges ahead. Any questions in the floor? Questions? Yes, please.
Artificial intelligence.
Thank you so much for your wonderful talk. My name is Ryuji Hamamoto from National Cancer Center, Japan. I completely agree with that epigenetics plays a critical role for biomarker of pancreatic cancer. So I would like to ask you the possibility of epigenetic changes as a target of therapy. So the very, very heterogeneous mechanism of pancreatic cancer. So do you have any plans to target epigenetics as a therapy?
Thank you very much, because here in the audience we have Professor Lomberg, who is the head of the experimental therapeutics branch of the center. Yes, all our experiments and experimental therapeutics is combining and using epigenomic drugs. There are wonderful publications from the Lomberg's laboratory and team on that. And we also have collaborated with other groups around the world to design therapies, and it is very exciting. I mean, actually extremely exciting. And I'm going to tell you why it is exciting. Because actually, as you know, pharmaceutical companies take a very easy route, and so they have taken for many years the enzymatic inhibitors. That is the approach, the enzymatic inhibitors and the enzymatic inhibitors. 7 years or 10 years ago, when I was consulting with companies on what it was going to be this decade, 60% of them, 68% of them chose as R&D primary epigenomic drugs. So you have now PRMT5 inhibitors in clinical trials, you have HDAC in clinical trials, etc. Now, I'm going to tell you one thing, and I'm going to be honest, and I'm going to repeat Josie again, who taught us a lot today. We create drugs. We discover drugs, and we do experimental therapeutics. But it is in the art of the practitioner and the fight of the patient and the family that make the drugs a miracle. So yes, we are very excited with epigenetic being at the center, and we are very excited that it also is a way in which the environment attacks you. So if you are telling me that you're getting a cancer for chronic pancreatitis, and you cannot get a gene, why are you getting a cancer for? Because you get a cancer from epigenetics. So it is inheritance, and that was something that very early on called us for that revolution. Now, very early on, we understood what you said. If we do not start, and that was when Dr. Lomberg joined us, and then she started with experimental therapeutic program, which is around 15 years old now, and it's very successful.
Okay, question online. How do you choose the KRAS mutants to include in your analysis? Briefly, I know it can be complicated.
Very briefly. We took every single publications of KRAS that existed all across. NIH has 88 mutations that they were doing, and we did 65. So there are the most common mutation, not only in the cancers, but also in something that maybe the audience is not that familiar, but I do study, and it helps me a lot to understand pancreatic cancer. And it's genetic diseases inherited that are called RASopathies. I don't know if you know them. There are many children walking with RASopathies, KRAS mutations that develop cardiac disease, for instance, and no cancer. What I learned about KRAS is that it was called, rightfully so, the proto-oncogene. Because KRAS, the way I see a pancreas full of, I'm almost ready to I'm writing this, so I hope that I can publish this. But my revolutionary thought is that I don't see the preneoplastic pancreas as a cancer. I see it as a rasopathy. And so we can treat it. We can treat the pancreas by the gastroenterologist, by the oncologist, as a rasopathy to begin with. And that will be one of the revolutions. Thank you very much.
Okay, and just wondered if there's another question from the floor. Yes, please.
Thank you very much, George Zagopoulos and Stephen Gallinger from Canada. Really enjoyed your talk. Fascinating work showing the heterogeneity among different KRAS mutations. Without being a reductionist, I want to come back to the genome and the genomic instability that probably gives rise to a lot of the epigenomic phenomena that you described that lead to progression and initiation progression. How about KRAS imbalance? As a KRAS imbalance as a driver over the heterogeneity that you're seeing with the number of mutants that you describe, given that in pancreas cancer, not as many mutants are driving the majority of the disease.
And speaking right into the microphone.
Yeah, speaking right on the microphone. Thank you for the question. It's a it's an active area of thought, actually, the imbalance, because there are different types of imbalance, as you might know. For instance, you have cancer in which you have more than one KRAS mutation, so you have different KRAS that you're going to be attacking in the same tumor. That is one RAS imbalance. The others are RAS imbalances that occur just by the same epigenetics mechanisms, for instance, and expressions. I think that the RAS imbalance in the biology of pancreas conceptually can influence the biology, but I actually, it's very hard to define what it will be the transforming potential of those imbalances. Where we are concerned is in the therapy, and that's where we will be diving more than in the biology. How can they be differentiated when they're coming to the imbalance?
Okay, thanks very much. Unfortunately, I'm sure there are lots of questions. If it's a quick one, if it's a very quick one, we can fit one more question in.
I'm not sure it's a quick one, but I'm not sure the response will be quick.
We will try.
Just a question around the resistance path. You did mention the fact that there was going to be heterogeneous resistance to KRAS inhibitors, for example. I think that's the case for every new targeted therapy that we're developing. How can we work together better earlier to try and predict those and help each other predicting those as soon as we've identified the targets? Because it's like chess, we really need to plan 3 or 4 shots ahead. So G7 cancer could be a good way for us to discuss that collaboration as well.
Yeah, I was going to make a joke first. How can we do, let's read each other's paper and let's accept each other's paper, let's move on. But I actually think that what I was referring is, I think I can explain it this way. If you have 25 KRAS or 65, 7 years ago, anyone would have told you that they're identical. Okay, that you have KRAS mutation, that's it, period. And that we made the G12 animal and that's it, right? So we were into that. Most of what we know of KRAS is G12, period. Okay, and so I think that the scenario that I was bringing is that if you develop if you are putting a pan inhibitor right now, okay, you're going to have different mechanisms because there are different signaling. And so there will be different mechanisms of target efficiency in targeting. But obviously that escape will be different because you have GR, for instance, that is completely dependent of AKT that doesn't bind to NF1 versus G12D that is completely dependent of ERK pathway. And so the mechanisms that are going to emerge as you are shutting down are going to be different. And one might end up being the mTOR pathway that we will have to attack later, right, versus the other. So that's what I was referring, that we have to be very careful right now because we have a new weapon and we are very excited. And what I would like to do is to communicate the beautiful news of what we have, but also to communicate, let's be careful, now that we have these gold things, let's not put a bad name on it, right? Most of the drugs that receive a bad name were because they were using wrong. So let's put a stop, and we need more data on KRAS in order to, we are using the therapy, but we need more data on KRAS itself.
Professor Urrutia, thank you very much. We had another question online about KRAS mutations making it complicated when it comes to personalized treatment. That is the next keynote address. Professor Urrutia, thank you very much. Thank you very much.
Thank you.